Have you ever paused, really paused, to appreciate the incredible engineering marvel that is your own body?
It's pretty amazing when you stop to think about it.
Right. Think about it. How does our skin stretch and then flawlessly snap back into place after, a pinch or a pull?
Uh-huh.
How do our bones resist crushing forces, allowing us to jump, run, or lift heavy objects without crumbling?
The stresses they handle are incredible.
Exactly. And how do our lungs expand so effortlessly with every breath, then recoil like, I don't know, perfectly tuned springs? These aren't just squishy passive tissues.
Not at all.
They are built with an astonishing level of precision, microscopic architectural elements working in concert, forming this dynamic structure that constantly impacts and endures.
It really is architecture at a tiny scale.
Today, we're taking a deep dive into the unsung heroes behind much of this biological resilience: fibrous proteins.
The real structural workhorses.
Specifically, we're going to be talking about two absolute powerhouses: collagen and elastin.
Two crucial ones, definitely.
Our mission today isn't just to introduce you to these proteins. We're going far beyond the basics.
We want to get into the nitty-gritty.
Exactly. Our goal is to peel back the layers and truly understand the intricate details of these fundamental biological architects, from their tiniest building blocks, the amino acids,
Right down to the atoms.
to the staggeringly complex ways they are synthesized, organized, and critically, what happens when things inevitably go wrong.
Because things do go wrong sometimes.
Think of this deep dive as your shortcut to truly mastering the information about these essential components of the extracellular matrix, giving you the insights of someone at, well, the elite level in this field.
That's precisely right. And to truly grasp the significance of collagen and elastin, we first need to understand their home, the extracellular matrix, or ECM.
Okay, the ECM, what exactly is that?
Well, imagine the cells in your body as individual bricks. The ECM isn't just the mortar holding them together.
Right.
It's the entire scaffolding, the foundation, and even like the communication network outside of those cells.
So it's much more than just filler.
Oh, absolutely. This intricate three-dimensional network of macromolecules provides essential structural support, it anchors cells in place, and it plays a vital, active role in almost every cellular process, from growth and migration to differentiation.
Wow, okay.
And collagen and elastin are prime examples of the structural fibrous proteins within this matrix. They are distinctly different from what we call globular proteins.
Globular proteins, those are the more compact ones.
Exactly. They're typically compact, folded into spheres, and often function as enzymes, transporters, or signaling molecules inside cells. Collagen and elastin are long and stringy, fibrous.
Built for structure.
Precisely. And what's truly remarkable about them is that their unique mechanical properties, collagen's immense strength, or elastin's rubber-like elasticity, are a direct result of their specific amino acid compositions.
The building blocks again.
And the very precise ways those amino acids arrange themselves into sophisticated secondary and tertiary structures. It's molecular engineering at its absolute finest, really. A testament to nature's incredible design principles.
Okay, let's unpack this and start with the undisputed heavyweight champion, collagen.
Good place to start.
This isn't just some obscure protein you might vaguely remember from a biology class. It is unequivocally the most abundant protein in the entire human body.
By far. It's everywhere.
So if we were to zoom into the molecular level, what exactly is collagen? How does it look? And what makes it so incredibly common and versatile?
Well, at its core, a typical collagen molecule is a long, rigid, rope-like structure.
Like a rope.
Exactly. Picture three individual polypeptide chains, which we call alpha or IO chains. These aren't just lying next to each other. They're wound tightly around one another in a distinctive right-handed triple helix.
A triple helix. Okay,
Yep.
so 3 strands twisted together.
Right, so imagine a perfectly braided super rope, exceptionally robust and resistant to stretching. This foundational triple-helical structure is what gives collagen its incredible tensile strength, its ability to resist being pulled apart.
And what's absolutely fascinating is how this fundamental rope-like structure then adapts to perform vastly different functions in different parts of the body.
It's amazing, isn't it?
It's like a single building material that can be shaped and organized in a million different ways depending on the architectural need. Can you give us some examples of how collagen's form dictates its function across our tissues?
Absolutely. This adaptability is one of collagen's most remarkable features. While that basic triple helix is constant, its specific organization varies dramatically.
Okay.
For instance, in the general extracellular matrix that surrounds many of our cells, or maybe in the vitreous humor of your eye, that clear jelly-like substance that fills your eyeball, collagen is dispersed as a loose gel. Here, its role is to provide foundational support and maintain shape, allowing for some flexibility while still offering a structural scaffold.
So like a supportive mesh.
Sort of, yeah. A bit like the gelatin in the Jell-O mold. It holds things together, but it's not rigid.
Okay.
But then consider your tendons. Tendons are those incredibly tough, fibrous cords that connect your muscles to your bones.
Right, like the Achilles tendon.
Exactly. In tendons, collagen is organized completely differently. Here, it's bundled together in very tight, perfectly parallel fibers.
All lined up.
Perfectly lined up. This linear, highly ordered arrangement is what gives tendons their immense tensile strength, allowing them to withstand incredible pulling forces. Think about the stresses on your Achilles when you jump or run.
Makes sense. Designed for pure strength in one direction.
Pure, unyielding strength, almost like a high-tension cable. Now let's go to the cornea of the eye.
The clear part at the front.
Right, that transparent front part that covers your iris and pupil. Here, the collagen fibers are stacked in a highly organized, layered fashion, almost like perfectly arranged plywood sheets.
Plywood, that's the interesting comparison.
It helps visualize the layers. This precise stacking is absolutely critical because it allows light to transmit through with minimal scattering, giving you perfectly clear vision.
So the organization is key for transparency.
Absolutely. Even the slightest disruption in this arrangement can lead to cloudy vision. And finally, in bone.
Okay.
Collagen fibers are arranged at angles to each other, forming a crisscross pattern, much like the rebar in reinforced concrete.
Like rebar. Okay, I get that.
Yeah, this unique, multi-directional organization is what allows bone to resist mechanical shear from multiple directions, making it incredibly robust and able to withstand the very stresses of movement, impact, and weight-bearing from all angles.
Wow.
So what we're truly seeing here is nature's equivalent of a molecular toolkit, where a single base material, collagen, is tweaked in subtle yet profound ways to become rebar in one place, a transparent window in another, and a powerful cable elsewhere. It's an incredible testament to biological efficiency and adaptability.
That's incredibly insightful. It truly highlights collagen's versatility. And you mentioned types of collagen. It's not just one uniform substance, is it?
No, definitely not.
We're talking about a whole superfamily, over 25 identified types, plus other proteins that share collagen-like domains. So what differentiates these types, and how do these subtle variations lead to such diverse functions?
That's a critical distinction. The classification of collagen into different types is based on subtle yet functionally significant variations in their alpha chain composition and how they organize into larger structures.
Alpha chain composition, so the specific strands in the rope.
Exactly. While individual alpha chains are all roughly similar in size, around 1,000 amino acids long, it's the differences in their specific amino acid sequences that confer their distinct properties.
Okay.
For example, type I collagen, the most abundant type we just discussed, is typically made up of 2 I1 chains and 1 IA2 chain, often denoted as I101.
2 alpha 1s and 1 alpha 2.
Right. In contrast, type 2 collagen is composed of 3 identical IA1 chains, or
Alpha 1s.
These seemingly small differences in their fundamental building blocks lead to profound functional distinctions in the tissues where they reside.
So how are they grouped? You mentioned organization.
Right. We can broadly organize collagens into 3 main functional groups. First, and perhaps most familiar, are the fibril-forming collagens, which include types 1, 2, and 3.
Fibril-forming, those are the classic rope-like ones.
Those are the classic rope-like structures. When you look at them under an electron microscope, you can see these characteristic banding patterns along the linear polymers of fibrils.
Banding patterns, like stripes.
Exactly like stripes. These bands are a direct reflection of the incredibly regular, staggered packing of individual collagen molecules within the fibril, almost like perfectly aligned bricks. This staggered arrangement is absolutely key to their immense strength and stability.
Makes sense. So types 1, 2, and 3, where do we find them?
Okay, type I collagen, as we said, forms fibers found in supporting elements requiring very high tensile strength. Think tendons, ligaments, the dermis of your skin. They all endure immense pulling forces.
And bone, you mentioned bone earlier.
And bone, absolutely, and the cornea too. Type 2 collagen fibers, on the other hand, are restricted primarily to cartilaginous structures, like the cartilage in your joints or your nose.
So more for cushioning.
Resilience and cushioning, exactly. And type 3 collagen is prevalent in more distensible or stretchy tissues,
Like blood vessels.
like the walls of blood vessels, yes. Also the skin, especially in early wound healing, and the intestines. Its presence allows for elasticity and expansion in these dynamic tissues.
Okay, so fibril-forming. What's the next group?
The second major group is the network-forming collagens, primarily types 4 and 8.
Network forming, so not ropes.
Right. Unlike the fibril-forming types, these don't create distinct rope-like fibrils. Instead, they form an intricate three-dimensional mesh or network.
Like a net.
Pretty much. Type 4 collagen is particularly important here, as its molecules assemble into sheet-like meshworks that constitute a major part of basement membranes.
Basement membranes, I've heard of those. They're like thin sheets under cells.
Exactly. These are incredibly vital, thin, sheet-like structures found throughout the body, providing crucial mechanical support for adjacent cells and acting as semi-permeable filtration barriers.
Filtration barriers, like in the kidneys.
Perfect example.
Yeah.
Imagine their function in your kidneys, where they selectively filter macromolecules from your blood or in your lungs, supporting the delicate alveolar structures critical for gas exchange. Their mesh-like structure allows for that selective passage.
Fascinating. Okay, fibril forming, network forming. Is there a third group?
Yes. Finally, we have the fibril-associated collagens with interrupted triple helices, often abbreviated as facet collagen.
Nasty seeds. Okay, that's a mouthful.
It is. These include types 9X and 12. Their role is quite specific and acts as a sort of molecular glue.
Glue?
Yeah, they bind to the surface of the larger classic collagen fibrils. In doing so, they link these fibrils to one another and also connect them to other components within the broader extracellular matrix.
So they help organize everything.
Exactly. They essentially help to organize the entire scaffolding system, integrating different structural elements and ensuring the tissue is a cohesive functional unit. So while all are collagen, their specialized forms ensure they can perform a wide array of very specific structural jobs, all tailored to the precise mechanical demands of the tissue they inhabit.
It's a true masterpiece of biological specialization.
It really is.
Here's where it gets really interesting for me. We've talked about these incredible structures, from ropes to networks, and how they contribute to our body's amazing resilience. But how do these proteins achieve such specific and robust structures at the molecular level, right down to their fundamental building blocks? How does the cell ensure that each brick is perfectly shaped and placed?
That's the absolute core of the mystery, and it's truly a marvel of biochemistry. The molecular structure of collagen is unique, especially when you compare it to those compact globular proteins we mentioned earlier.
Right, the spherical ones.
Collagen has this elongated triple-helical structure stabilized by extensive interchain hydrogen bonds.
Hydrogen bonds. Like tiny magnets holding the strands together.
Sort of, yeah. They literally hold the three strands together, like molecular Velcro. But the magic, as you said, begins with its amino acid sequence. Collagen is exceptionally rich in two very specific amino acids, proline and glycine.
Proline and glycine again.
These aren't just abundant by chance. They are absolutely critical for forming that signature triple-stranded helix.
Oh, so what do they do?
Well, proline, for example, has this distinctive ring structure. When it's incorporated into the polypeptide chain, it introduces what we call kinks, or tight bends.
Kinks? Doesn't that sound bad?
Not here. It's actually crucial because it facilitates the helical conformation of each individual alpha chain, preventing it from forming the more common, wider alpha helix that you see in many other proteins. So proline gives each strand its initial precise coil.
Ah, okay, so the kink helps it twist correctly.
Precisely. Then there's glycine. Glycine is the smallest amino acid imaginable. Its diminutive size is its superpower here. It fits perfectly into the incredibly restricted, cramped spaces where the three chains of the triple helix come together at the very core of that rope-like structure.
So only glycine can fit in the middle?
Basically, yes. If it were any larger, even slightly, the chains simply couldn't pack so tightly and form that stable triple helix.
Wow.
This essential pairing leads to a very characteristic repeating sequence you'll find throughout collagen.
GlyXY.
GlyXY, right. In this repeating pattern, X is very frequently proline, and Y is often hydroxyproline, or less commonly, hydroxylation.
Wait, hydroxyproline, hydroxylation, are those standard amino acids?
Good question. They are non-standard because they result from post-translational modification.
Post-translational, meaning after the protein chain is made.
Exactly. Hydroxylation adding an OH group happens to proline and lysine residues after they've been incorporated into the polypeptide chains.
Okay, so why hydroxyproline? Why add that OH group?
Crucially, hydroxyproline maximizes the formation of those interchain hydrogen bonds we mentioned.
The molecular Velcro.
Right. Those hydrogen bonds are what stabilize the triple helical structure. More hydroxyproline means more hydrogen bonds, means a much stronger, more stable helix.
Got it. Stability.
And there's a really important cofactor needed for this hydroxylation reaction.
Oh, what's that?
These hydroxylating enzymes, proline hydroxylase and lysyl hydroxylase, they need molecular oxygen, ferrous iron, FeO, and critically, vitamin C ascorbic acid.
Vitamin C, seriously? Like the vitamin in oranges?
The very same.
Yeah.
Without vitamin C, these hydroxylating enzymes simply cannot function. They stop working.
And what happens then if you don't have vitamin C?
Well, this is where we see the direct, undeniable vitamin C connection to a debilitating disease. Scurvy.
The
Right?
disease sailors used to get.
Exactly. If there's a deficiency of vitamin C, the hydroxylation of proline and lysine is severely impaired. This, in turn, critically limits the formation of those essential interchain hydrogen bonds.
So the helix isn't stable?
Precisely. The result: an unstable triple helix and subsequently impaired cross-linking of the collagen fibrils once they are secreted into the extracellular space. This drastically decreases the tensile strength of the assembled collagen fiber.
Which explains the symptoms of scurvy.
Absolutely. Patients with scurvy often exhibit symptoms like ecchymosis, those large bruise-like discolorations on the limbs,
Right.
which are actually due to subcutaneous extravasation or leakage of blood from incredibly fragile capillaries just beneath the surface of the skin. Their blood vessels literally become weak because the collagen isn't strong enough.
Wow. It's a vivid demonstration of how critical that one vitamin is at a molecular level.
It really is. It's why sailors centuries ago would literally watch their bodies fall apart, all because this essential molecular step was missing.
Okay, so back to the assembly line. We've got hydroxylation. What else happens in the RER?
Right, returning to the RER, in addition to hydroxylation, some of those newly formed hydroxylation residues are also enzymatically glycosylated.
Glycosylated, meaning sugars are added.
Yes, typically glucose and galactose are added. This crucial sugar addition happens before the triple helix forms, adding another layer of modification.
That's a lot of intricate steps just for the building blocks. So once the cell has these precisely structured individual chains, how does it actually make these massive, complex collagen fibers? It sounds like an incredibly orchestrated assembly line.
It absolutely is. It's a multi-step, highly coordinated process that begins inside the cell, proceeds through various cellular compartments, and culminates in the vast extracellular matrix outside the cell. Let's walk through this.
Okay, lay it out for us.
First, we have pro-8 chain formation. Collagen is an exported protein, meaning it functions outside of the cell.
Right, in the ECM.
Like other proteins destined for secretion, its newly synthesized polypeptide precursors, initially called pre-pro chains, contain a special signal sequence at their amino N-terminal end.
A signal sequence, like a zip code.
Exactly like a zip code or maybe a shipping label, it acts like a molecular GPS, directing the ribosomes, where the polypeptide is being synthesized, to bind to the rough endoplasmic reticulum, or RER. It then threads the growing polypeptide into the RER lumen, which is the space inside the RER. Once inside, that signal sequence is rapidly cleaved off by an enzyme, leaving us with a pro-8 chain now ready for further modifications.
Got it. Signal removed, chain is inside the RER, then comes the hydroxylation and glycosylation we just talked about.
Precisely. Within the RER lumen, while the polypeptides are still being synthesized, those crucial hydroxylation and glycosylation reactions occur. Prolines and lysines get hydroxylated if vitamin C is present, and some hydroxylations get sugars attached.
Okay, chains are modified. What's next?
Once these modifications are complete, the next major step is procollagen assembly and secretion. 3 of these modified pro chains come together to form procollagen.
Trocollagen, so almost collagen, but not quite.
Exactly. This is a precursor molecule that has that central triple-helical region, but it's flanked by non-helical N and C terminal extensions called propeptides.
Oh, peptides. What are they for?
They're actually really important at this stage. They help guide the assembly and prevent the collagen from forming large fibers inside the cell, which would be disastrous. What's truly clever here is that the formation of procollagen actually begins with the precise formation of interchain disulfide bonds between the C-terminal extensions.
The sulfide bonds, sulfur bridges.
Yes, between the C-terminal peptides of the procuzet chain. This effectively pulls the 3 alpha chains into the perfect alignment needed for the triple helix to form correctly, ensuring it zips up perfectly from the C-terminus towards the N-terminus.
So the ends hook up first to guide the zippering. That's smart.
Very smart design. Once assembled, the procollagen molecules then move from the RER through the Golgi apparatus.
His cell's post office.
Sort of, yeah, where they're further processed and packaged into secretory vesicles. These vesicles then travel to the cell membrane, fuse with it, and release the procollagen molecules into the vast extracellular space.
Okay, so procollagen is now outside the cell. Is it collagen yet?
Not quite. There's a vital step of extracellular cleavage. Once outside the cell, the triple-helical procollagen molecules are precisely cleaved by enzymes called N and C procollagen peptidases.
Peptidases.
Protein cutters? Exactly. These enzymes act like molecular scissors, specifically removing those terminal propeptides, while those propeptides were absolutely essential inside the cell.
For guiding assembly and preventing premature clumping.
Right. They are no longer needed once the molecule is outside the cell. What remains are the tropocollagen molecules. These are the true structural units ready for assembly into larger structures.
Tropocollagen. Okay, now we're getting somewhere. What happens to them?
These tropocollagen molecules then spontaneously self-assemble into collagen fibrils.
Spontaneously? They just do it on their own?
Pretty much, based on their shape and chemical properties. They arrange themselves in a very ordered parallel array, but it's a staggered pattern. Each tropocollagen molecule overlaps its neighbor by about 3/4 of its molecular length.
The staggered bricks again.
Exactly. This precise staggered packing is what gives collagen fibrils their characteristic banding pattern seen under an electron microscope and contributes significantly to their strength, making them resilient to pulling forces.
Okay, so they line up into fibrils. Is that the final step?
Almost. For true strength and long-term stability, we have cross-link formation. This beautifully arrayed fibrillar structure of collagen molecules now becomes the substrate for an enzyme called lysyl oxidase.
Lysyl oxidase. I feel like we've heard that name before.
We will again. It's a crucial copper-containing enzyme that operates extracellularly. Lysyl oxidase oxidatively deaminates some of the lysine and hydroxylation residues present in the collagen.
Meaning it chemically modifies them.
Yes, it removes an amino group and creates a reactive aldehyde group. This reaction produces highly reactive aldehydes called allicine and hydroxylacylane.
Allicine, okay.
These aldehydes then spontaneously condense with other lysine or hydroxylacylane residues on neighboring collagen molecules, forming incredibly strong, stable, covalent crosslinks.
Covalent bonds. The really strong chemical
The
bonds.
strongest kind. These robust crosslinks are what truly superglue the collagen fibrils together, creating mature, stable collagen fibers with remarkable tensile strength, ensuring they don't unravel under stress.
Wow. That final cross-linking step sounds critical.
Absolutely critical.
But
As a powerful clinical aside, it's worth noting that if lysyl oxidase is inhibited, for example, by a toxin found in sweet pea seeds,
sweet peas, really?
yes, it can lead to a condition called latherism, characterized by severe skeletal deformities and vascular problems. It highlights just how critical this enzyme is for structural integrity.
So problems with lysyl oxidase mean weak collagen.
Very weak. And lysyl oxidase is just one of several copper-containing enzymes in the body.
Copper, like the metal.
Exactly. Others include ceruloplasmin, cytochrome C oxidase, superoxide dismutase. This means that disruptions in copper homeostasis, how the body handles copper, as seen in genetic disorders like Menkes syndrome, a deficiency, or Wilson disease, an overload,
Right.
can have far-reaching and devastating effects due to the impaired function of these crucial copper-dependent enzymes, affecting everything from energy production to antioxidant defense and, of course, connective tissue formation.
So copper levels are really important for connective tissue too.
Very important.
Okay. So these incredibly stable, super strong fibers are painstakingly built step by step, right from the amino acid level up to the full fiber, a true feat of biological engineering. But our body isn't static. It's constantly remodeling, repairing, and growing.
Absolutely. Constant turnover.
We replace old cells, heal wounds. How does something so meticulously built and stable actually get broken down efficiently when it's no longer needed or if it's damaged?
That's a great perceptive question, and it speaks to the dynamic nature of our connective tissues. While mature collagen molecules are indeed highly stable, with half-lives that can span several years.
Years. Wow.
Some collagen in your body today could have been there for a decade. but the connective tissue itself is continuously being remodeled. This remodeling is absolutely vital, often occurring in response to growth, repair after injury, or simply the normal wear and tear of daily life.
So how does the breakdown happen?
The breakdown of these robust collagen fibers relies on the proteolytic action of a very specific group of enzymes called collagenases.
Collagenases, enzymes that break down collagen. Makes sense.
Exactly. These enzymes belong to a larger family known as matrix metalloproteinases, or MMPs.
MMPs.
They
Okay.
are essentially molecular demolition crews.
Mm.
For the most common type I collagen, the
Mhm.
cleavage by collagenases is remarkably precise, producing distinct fragments. One exactly 3/4 of the molecule's length and one precisely 1/4 length.
A very specific cut.
Very specific. These initial fragments are then further degraded into smaller peptides and amino acids by other matrix proteinases, ensuring that the components can be recycled or cleared, allowing for constant renewal and adaptation of the tissue.
So it's a controlled demolition and recycling process.
Perfectly put, this dynamic balance between synthesis and degradation is crucial for maintaining tissue health and integrity.
So what does this all mean for us? We've just explored this incredibly precise, multi-step building process for collagen, a marvel of biological engineering. Every single step, from the right amino acids to the correct enzymatic cuts and cross-links, seems to have to go perfectly.
The tolerances are incredibly tight.
But what happens when there's a flaw in those blueprints? The genes or if the construction crew, the enzymes, make a critical mistake at any stage.
That's when we encounter collagenopathies. These are a group of often debilitating genetic diseases that arise from defects in collagen fibrosynthesis, assembly, or breakdown.
Collagenopathies?
Diseases of collagen. Right. They essentially mean the body's ability to form collagen fibers properly is impaired, leading to a failure to provide tissues with the necessary tensile strength that collagen normally provides.
And these are genetic?
Primarily genetic, yes. What's truly astounding is the sheer number of known issues. Over 1,000 different mutations have now been identified across 23 genes that code for just 13 of the collagen types.
1,000 mutations. That's incredible.
It highlights the vast complexity, the sheer precision required, and the critical importance of every single step in the process. A single misstep can have profound, widespread consequences throughout the body.
Can you give us some examples?
Absolutely. Let's look at two prominent and impactful examples. The first is Ehlers-Danlos syndrome, or EDS.
EDS. I've heard of that. It affects joints, right?
Joints, skin, blood vessels. It's actually not a single disease, but a heterogeneous group of connective tissue disorders that are inherited, resulting from various heritable defects in the metabolism of fibril or collagen molecules.
So different types of EDS have different causes.
Exactly. The causes are varied. It can stem from a deficiency of enzymes crucial for collagen processing, such as lysyl hydroxylase or N-procollagen peptidase.
So the construction crew isn't working properly.
Right. Or it can be due to specific mutations in the amino acid sequences of collagen types I, 3, and V themselves.
Meaning the blueprints are faulty.
Precisely.
So what are the typical symptoms?
Well, the classic form of EDS, which is often linked to defects in type V collagen, is characterized by remarkably extensible and fragile skin.
Stretchy skin.
Very stretchy. Imagine skin that can stretch far more than normal, almost like chewing gum, but is then prone to tearing, bruising, and poor wound healing. This is combined with significant joint hypermobility.
Super flexible joints.
Right, where individuals can bend their joints in seemingly impossible ways, leading to frequent dislocations and chronic pain.
This sounds really difficult to live with. Is there a more dangerous form?
Yes, unfortunately. The vascular form of EDS, caused by defects in type 3 collagen, is by far the most serious.
Why is that?
This is because it's associated with a potentially lethal risk of arterial rupture, as the blood vessel walls, which rely heavily on type 3 collagen for their distensibility and integrity, become dangerously fragile and prone to tearing.
Wow, that's incredibly serious.
It is. And what's particularly challenging with many collagenopathies like EDS is the concept of a dominant negative effect.
Dominant negative, what does that mean?
It means that if even just one mutant collagen chain, produced from one faulty gene copy, is incorporated into a triple helix, it can disrupt the entire structure, acting like a bad apple that spoils the whole barrel.
So one bad chain ruins the whole molecule.
Exactly. This single faulty chain can lead to misfolding and rapid degradation of the entire molecule, even if other normal chains are present. It's like having one faulty brick that undermines the structural integrity of the entire wall, causing it to collapse.
That explains why even inheriting one bad copy can cause severe problems.
Precisely.
Okay, EDS is one example. What's the other major one?
Our second example is osteogenesis imperfecta, OI, more commonly known as brittle bone disease.
Brittle bone disease, that sounds self-explanatory.
It is tragically descriptive. This is a devastating genetic disorder characterized by extreme bone fragility, where bones can fracture easily with only minor or even no trauma at all.
Like breaking a bone just rolling over.
It can be that severe, yes. Imagine a child breaking a bone just by rolling over in bed. Over 80% of OI cases are caused by dominant mutations in the genes that encode either the ADA1 or ADA2 chains of type I collagen.
Type I collagen, again, the main one in bone.
Exactly. The very collagen we discussed is providing high tensile strength, especially in bones. The most common mutations involve the replacement of glycine.
Glycine, that tiny amino acid that has to fit in the middle.
That's the one. Replacement of glycine in the glycine XY sequence with larger, bulkier amino acids. This seemingly small change has catastrophic consequences because that larger amino acid physically prevents the crucial triple-helical conformation from forming correctly.
It just doesn't.
It just doesn't fit, leading to unstable and weak collagen.
And I assume there are different severities of OI.
Yes, the severity of OI, or its phenotype, ranges dramatically from very mild to lethal. Type IOI is the most common and generally mildest form, characterized by fragile bones, some hearing loss, and a distinctive symptom: blue sclerae.
Blue sclerae, blue whites of the eyes.
Exactly. The sclera are the whites of your eyes, and in OI they can appear blue due to the thinning of the connective tissue, which allows the dark underlying choroid layer, rich in blood vessels and pigment, to show through.
Wow. Okay, that's type I. What about the more severe types?
Type 2 OI is the most severe form and is often lethal in the perinatal period, meaning around the time of birth,
That's awful.
primarily due to severe pulmonary complications, as the rib cage and lungs cannot form properly. Tragically, multiple in utero fractures are frequently observed in these cases, and newborns often present with a crushed appearance.
Oh my goodness.
Type 3 OI is also a severe form, characterized by multiple fractures at birth, significantly short stature, and severe spinal curvature, leading to a humped back or kyphotic appearance, again, often accompanied by blue sclerae.
So multiple fractures right from birth.
Yes. And some individuals with OI might also experience dentinogenesis imperfecta.
Problems with teeth.
Yes, a disorder affecting tooth development, leading to discolored, brittle teeth. It's worth noting, too, that blue sclerae can be a shared symptom across several connective tissue disorders, including Marfan syndrome, OI, and EDS.
Because of the thin tissue allowing the layer underneath to show through.
Exactly. It underlines how fundamental these proteins are to virtually every system in our body. A problem here ripples outwards.
Incredible. Okay, let's shift gears now and talk about collagen's brilliant counterpart, elastin.
Elastin, yes, the stretchy one.
If collagen provides the tough, unyielding tensile strength, acting like the rebar in a building, then what exactly is elastin's superpower? What unique role does it play in our tissues, and how does it accomplish that?
What's fascinating here is that elastin provides precisely the opposite yet equally vital property: rubber-like elasticity.
Elasticity. Makes sense, given the name.
Right. While collagen provides strength and resistance to stretching, elastin allows tissues to stretch several times their normal length and then recoil perfectly back to their original shape when the stretching force is relaxed.
Like a biological rubber band.
The ultimate biological rubber band, engineered for constant movement and resilience. Its primary locations in the body highlight this function. It's incredibly abundant in the lungs.
For breathing.
Exactly, allowing them to expand and contract effortlessly with each breath. It's in the walls of large arteries like the aorta.
Handling blood pressure changes.
Crucial for accommodating rapid blood pressure changes during each heartbeat and maintaining smooth blood flow. And it's in elastic ligaments, which need to stretch and return to their original shape.
Like in the spine.
Yes, like the ligamentum flavum in the spine, which helps us bend and straighten.
So structure-wise, how is elastin different from collagen?
Let's delve into the structure of elastin. It starts its life as a soluble polypeptide precursor called triple-elastin.
Triple-elastin. Okay.
This molecule is roughly 700 amino acids long and is composed primarily of small, non-polar amino acids, such as glycine, alanine, and valine. This differs significantly from collagen's amino acid profile.
How so?
Well, while elastin is rich in proline and lysine, crucially, It contains scant amounts of hydroxyproline and hydroxylation.
So not much of the hydroxylated ones that stabilize collagen.
Exactly. This difference is key to its elasticity, as it avoids the rigid hydrogen bonding network that makes collagen so stiff it allows for more flexibility.
Okay, so triple-elastin is made. What happens next?
Triple-elastin is secreted by the cell into the extracellular matrix. There, it doesn't just spontaneously organize into functional elastic fibers like triple-collagen does. It needs help.
Help from what?
It interacts with specific glycoprotein microfibrils, primarily one called fibrillin.
Fibrillin. Okay.
Fibrillin acts as a vital scaffold, almost like a temporary guide rail, providing the structural framework onto which triple-elastin molecules are precisely deposited and assembled into nascent elastic fibers.
So fibrillin lays down the track for triple-elastin to follow.
That's a great analogy. This scaffolding is absolutely crucial for the correct organization and alignment of elastic fibers, ensuring they form a functional, resilient network. Without fibrillin's guidance, triple-elastin would just, well, clump together ineffectively.
Got it. Fibrillin scaffold triple-elastin deposits. Then what makes it elastic?
Once deposited, the magic of elastin's extreme elasticity comes from its unique and complex cross-linking. Similar to collagen, lysyl oxidase, that same copper-containing enzyme we discussed earlier, plays a critical role here.
Lysyl oxidase again. It's busy.
Very busy. It oxidatively deaminates some of the lysine side chains of triple-elastin, forming allicine residues just like in collagen. But here's the truly unique part. Instead of just two residues forming a cross-link, as in collagen and elastin, 3 allyl side chains, plus 1 unaltered lysyl side chain from the same or neighboring polypeptides come together.
4 chains involved in one link?
Yes. They form a distinctive, highly complex, and incredibly strong structure called a desmosine crosslink.
Desmosine, that's unique to elastin.
Absolutely unique to elastin. These desmosine crosslinks create an extensively interconnected, three-dimensional, rubbery network. It's this unique web of flexible and interconnected desmosine crosslinks that allows elastin to stretch and bend in any direction when stressed, and then spring back, giving connective tissue its remarkable elasticity and recoil properties.
So those desmosine links are like the connection points in a stretchy net.
Exactly, like having billions of tiny interconnected springs within your tissues.
And you mentioned fibrillin, the scaffold. Does anything go wrong with that?
Yes, absolutely. This intricate structure and the vital role of fibrillin also connect directly to another significant genetic disorder, Marfan syndrome.
Marfan syndrome, I know that affects height and heart, right?
That's right. Mutations in the fibrillin-1 protein, the very scaffold onto which triple-elastin is deposited, and which forms elastic microfibrils, are responsible for Marfan syndrome.
So the scaffold itself is faulty.
Precisely. This is a severe connective tissue disorder that dramatically impacts structural integrity throughout the body, particularly in the skeleton, leading to tall stature, long limbs, the eyes, leading to lens dislocation, and crucially, the cardiovascular system.
The heart issues.
Yes, where it can cause life-threatening aortic aneurysms and dissections, where the wall of the main artery tears. When the fibrillin-1 protein is abnormal, it disrupts the formation of functional microfibrils, leading to compromised, weak, and non-resilient elastic tissue throughout the body.
Because the foundation was flawed from the start.
Exactly. The fundamental scaffolding is flawed, so the elastic fibers built upon it are also flawed and weak.
Okay, so elastin provides incredible flexibility, allowing our tissues to stretch and recoil millions of times over a lifetime. But like all proteins, it's subject to breakdown and degradation.
It is, yes.
What protects this vital, irreplaceable elasticity from being broken down prematurely, especially in places like our lungs, where that continuous stretch and recoil is so vital for life?
That's where alpha-1 antitrypsin, AAT, comes in, playing the role of a crucial guardian.
Alpha-1 antitrypsin, AAT, what does it do?
AT is a potent protein found abundantly in our blood and other body fluids, and its primary function is to inhibit a number of proteolytic enzymes that hydrolyze and destroy proteins.
So it stops protein breakdown?
Certain kinds, yes. While it was initially named for its ability to inhibit trypsin, its most important physiological role, especially in the lungs, is to inhibit neutrophil elastase.
Neutrophil elastase, an enzyme that breaks down elastin from neutrophils.
Exactly. Neutrophil elastase is a powerful, aggressive protease that is released into the extracellular space, particularly by neutrophils, which are a type of white blood cell and key players in our immune system. This enzyme is highly capable of degrading elastin in the alveolar walls of the lungs, as well as other structural proteins in various tissues.
And AAT stops it. Where does AAT come from?
Mostly, yes. Most of the AT found in our plasma is synthesized and secreted by the liver, although some extrahepatic synthesis also occurs in other tissues.
So the liver makes this protective protein for the lungs.
Primarily, yes. This brings us to a delicate and critical balance, particularly in the lungs.
A balance between elastase and AT.
Precisely. In a normal, healthy lung, the alveoli, those tiny delicate air sacs where vital gas exchange occurs, are chronically exposed to low levels of neutrophil elastase.
Why? Why is elastase there normally?
It's constantly being released from activated and degenerating neutrophils, which are always present as part of the body's ongoing immune surveillance against airborne pathogens and irritants we breathe in.
Ah, okay. Part of the normal defense system.
Right. In a healthy individual, AAT is the primary inhibitor, acting like a constant sentry. It actively neutralizes this elastase, forming a stable complex with it and preventing it from destroying the precious elastin in the alveolar walls.
So AAT keeps the elastase in check.
Exactly. This balance is absolutely critical because lung tissue, once damaged by elastase, essentially cannot regenerate.
It can't heal itself.
Not the elastic structures, no. So the constant protection offered by AAT is paramount to preventing permanent, irreversible destruction of the connective tissue and alveolar walls, which would otherwise lead to severe pulmonary disease.
This raises an extremely important question. What happens if that protective balance is disrupted, particularly with AY1 antitrypsin? If our body's guardian against elastase isn't functioning properly, how does that lead to disease?
That's when we see the serious, life-altering implications of A1 antitrypsin deficiency and emphysema.
Emphysema, that's a severe lung disease.
It is. In the United States, roughly 2% to 5% of patients diagnosed with emphysema are predisposed to the disease because of inherited defects in their AET.
So it's a genetic cause for some emphysema cases.
Yes. While several different mutations in the AAT gene are known to cause a deficiency of the protein, one particular single purine-based mutation is clinically the most widespread and severe.
One specific mutation.
Yes, a specific change from J to AG in the genetic code, which results in the substitution of lysine for glutamic acid at position 342 of the AT protein. This mutated protein is commonly known as the Z variant.
The Z variant, okay. What's wrong with it?
What makes this Z variant mutation so problematic is that it causes the normally monomeric AAT protein, which means it functions as a single individual unit, to misfold.
Misfold, like it doesn't fold into the right shape.
Exactly. Instead of remaining as individual functional units that can be secreted from the liver, the Z variant AAT molecules polymerize and aggregate within the rough endoplasmic reticulum of the hepatocytes.
They stick together inside the liver cells.
Precisely. They get stuck. This means AT deficiency is essentially a misfolded protein disease, where the problem isn't necessarily a lack of production of the protein, but a failure of its proper folding and secretion. It gets trapped in the liver.
So if it's trapped in the liver, it can cause liver problems too.
Yes, exactly. This misfolding leads to a dual pathology, affecting both the liver and, importantly, the lung.
How does it affect each?
In the liver, the accumulation of these aggregated AAT polymers within the hepatocytes can lead to cellular damage and ultimately cirrhosis, which is scarring of the liver, potentially progressing to liver failure.
Okay, liver damage from the buildup and the lungs.
Simultaneously, because the misfolded AT is trapped and not efficiently secreted from the liver, there's a significant decrease in AT levels in the blood. Consequently, a drastically reduced amount of functional AAT reaches the lungs.
So the lungs lose their protection.
They lose their guardian. This deficiency allows neutrophil elastase to act unopposed, without its crucial inhibitor, leading to the relentless destruction of the alveolar walls and eventually pulmonary emphysema.
The breakdown of the air sacs?
Yes, a chronic progressive lung disease that makes it increasingly difficult to breathe as the air sacs are irreversibly damaged and lose their elasticity.
How is this inherited? Do you need one bad copy or two?
For an individual to be at significant risk for developing emphysema due to AAT deficiency, they must inherit two abnormal AAT alleles, one from each parent. The ZZ genotype.
Two copies of the Z variant. What if you only have one?
Individuals who are heterozygotes, meaning they have one normal M gene and one defective Z gene, MZ genotype, usually produce sufficient levels of AAT to protect their alveoli from damage, though they may have slightly lower levels than MM individuals. They are considered carriers.
Okay, so two bad copies cause the lung disease risk. Is there anything that makes it worse?
Yes. A critical factor that dramatically impacts the progression of emphysema in AAT-deficient individuals is smoking.
Smoking. I suspected that might come up. How does it interact?
It's a devastating synergistic effect. There's a specific amino acid, methionine 358, in AAT that is absolutely essential for the inhibitor to bind to its target proteases, like neutrophil elastase.
A key binding site.
Exactly. Smoking causes the oxidation and subsequent inactivation of this crucial methionine, literally changing its chemical structure. This renders the AAT completely powerless to neutralize elastase, even if some AAT is present.
So smoking poisons the AAT that is there.
Effectively, yes.
Yep.
Therefore, smokers who also have AAT deficiency, the ZZ genotype, experience a considerably elevated rate of lung destruction and have a significantly poorer survival rate compared to non-smokers with the same deficiency.
Wow, that's a stark warning about smoking, especially for those with the genetic risk.
Absolutely. It's a prime example of how environmental factors can profoundly exacerbate genetic predispositions.
Is there any treatment for AAT deficiency?
Fortunately, yes. There's a treatment available called augmentation therapy.
Augmentation therapy, what does that involve?
AAT protein, collected from the plasma of healthy human donors, is administered intravenously, usually on a weekly basis. This infused AAT diffuses from the blood into the lung fluid surrounding the lung epithelial cells.
Getting into the lungs where it's needed.
Right. It allows it to reach therapeutic levels and provide the much-needed protection against elastase, essentially replacing the missing or non-functional protein and restoring that crucial balance.
So we've taken a truly deep dive into collagen, the body's super strong rebar, providing incredible tensile strength and shaping everything from our bones to our eyeballs.
A master builder.
And we've explored elastin, its incredible stretchy rubber band, giving our lungs and arteries that vital life-sustaining flexibility.
The master of recoil.
We've peeled back the layers to see how precise molecular structures lead to vastly different functions, and how even tiny errors in their blueprints or their complex construction pathways can have profound, sometimes devastating health impacts.
It really drives home the precision needed.
This isn't just biology, it's an appreciation for the astonishing, intricate engineering within our own bodies.
If we connect this to the bigger picture, it's clear that understanding these fundamental fibrous proteins and their complex synthesis pathways isn't just an academic exercise confined to a textbook.
Right, it feels much more relevant than that.
It's about truly appreciating the incredible, meticulous design at the molecular level that underpins our very physical existence, our movement, our breathing, our vision, everything.
And knowing what can go wrong helps us understand disease.
Absolutely. Moreover, recognizing how these systems can go wrong highlights the critical importance of ongoing research into these molecular pathways. This research isn't abstract. It directly leads to the development of life-changing therapies.
Like the augmentation therapy for AT deficiency.
Exactly. Whether it's finding ways to treat brittle bone disease, repairing damaged connective tissue after injury, or protecting our lungs from chronic breakdown in conditions like emphysema, it truly informs how we approach health and disease at the most fundamental level.
What stands out to you from this deep dive, thinking back over everything we've covered?
It's a tough one. Maybe the sheer astounding complexity of protein synthesis, particularly collagen, that multi-step molecular factory.
The journey from inside the cell to outside.
Running perfectly inside every single cell, needing things like vitamin C. Or perhaps it's the devastating ripple effect of a single amino acid mutation like glycine in OI or the Z variant in AAT deficiency.
Yeah, how one tiny change can undermine an entire system. That really struck me too, the fragility despite the strength.
Exactly. That balance is key.
As you go about your day now, consider how our daily lives, the food we eat, the air we breathe, the movements we make, constantly interact with these microscopic architects, shaping the very resilience and structure of our bodies.
They're constantly working, constantly adapting.
What other hidden molecular wonders underpin our health that we often take for granted, humming along perfectly until something goes awry? Go forth and keep exploring.